Dear students, welcome to EPG Pathshala. Today I am here to present my module Anaerobic Digester. Anaerobic Digester, it is something to deal with or to work with in the absence of air. Anaerobic digester nowadays are providing promising and eco-friendly technology to the world. Why? Because it not only improving the environmental health, but it is also improving the human health by removing or by minimizing the greenhouse gases in the atmosphere, plus the odor is also being removed, and to somehow we are able to manage our organic waste also. The end product which is being formed in the form of gas, it is also being used for or as an alternate of energy. In the developing countries like ours, India, we are also using this technology, anaerobic digester technology, not only to manage our waste but also to use the energy which is being produced in the form of gas for various purposes like heating and cooking purposes. In anaerobic process or anaerobic digesters, we usually take organic waste like waste from food industries, waste from dairy industries, and these waste they are now being anaerobically digested in these digesters. In the digesters, the microorganisms they play a very important role in controlled conditions and decomposed our this organic waste into useful byproducts. Two kinds of byproducts we gain from the anaerobic digester. One is our biogas and another is in the form of liquid which can be used as a liquid fertilizer for our agriculture purpose. On the other hand, the biogas which is being generated can be used for electricity purpose, for heating, for cooking, etc. Now in the anaerobic digester, the microorganisms play their role in four different phases. First phase you can say is of hydrolytic phase, then comes your acidogenesis phase and then methanogenesis phase. During these phases, different microorganisms they act on our waste and decompose it to a end product like methane. In the anaerobic digestion process, we can use different kinds of digester like our floating drum, your fixed drum digester. The digester can be in tubular form, in garage form also we can use the digester. So these are the different kind of digester with which we can treat our organic waste. Now the digester, its economical value only increase when it generates or when we use that energy in the form of biogas which is being generated into a useful purpose. Now in the anaerobic digester, the microorganisms play their role in four different phases. First phase you can say is of hydrolytic phase, then comes your acidogenesis phase and then methanogenesis phase. During these phases, different microorganisms they act on our waste and decompose it to a end product like methane. In the anaerobic digestion process, we can use different kinds of digester like our floating drum, your fixed drum digester. The digester can be in tubular form, in garage form also we can use the digester. So these are the different kind of digester with which we can treat our organic waste. In this slide, anaerobic microbial digestion process has been explained. The first phase, that is your hydrolysis phase. In this phase, hydrolytic bacteria try to convert our complex organic substrate into simpler monomer units. For example, protein, carbohydrate, fats, these are now being converted into simpler units. In the second acidogenesis phase, acidogenic bacteria they plays very important role. Now the amino acids, sugars, fatty acids being produced during this phase. The third phase is acidogenesis phase. In this phase, all the amino acids, sugars and fatty acids which has been produced in the second phase act as intermediate substrate for acidogenesis phase, and the intermediate like propionic acid, butyric acid, it has been produced. Now the third phase is acidogenesis phase. In this phase, acetogenic bacteria, they plays a very important role, and the intermediates now are being converted into acetic acid or hydrogen. Methanogenesis phase, that is the important phase. In this phase, methanogenic bacteria, they plays a very important role, and they try to convert acetic acid and hydrogen into a valuable product, that is methane. In this slide, types of digester has been explained. Now the first we are taking is fixed dome digester. A fixed dome plant is composed of a closed dome shaped digester with an immovable rigid gas holder, as the name indicates, that is fixed dome digester. There is a feed stock inlet and a displacement pit compensation tank. The gas evolved in the digester is stored in the upper part of the reactor, and it is being collected from the gas outlet. Next is floating drum digester. A floating drum biogas plant composed of a cylindrical digester and a movable floating gas holder. That's why this digester has been given the name of floating drum digester. The gas produced can be accumulated in the gas drum, which rises or falls, again depending on the amount of gas produced and used. A well sustained metal gas holder can be supposed to last from 3 to 5 years in humid areas and 8 to 12 years in a dry climate. In this slide, the tubular digester has been explained. The tubular biogas plant, composed of a longitudinal shaped, heat sealed, weather resistant plastic or rubber bag, you can say that it is having a balloon shape that can be as a digester and a gas holder in one end. The gas is stored in the upper part of the balloon. The inlet and the outlet are attached directly to the balloon. The benefits are that their construction cost is very low. Also, shallow below ground installation makes them appropriate for use in areas with a high groundwater table. But the plastic balloons is quite breakable and vulnerable to mechanical damage and a very short life of 2 to 5 years. In this slide, garage type digester has been explained. In contrast to the above mentioned reactors, the garage type digester is operated in batch mode with dry digestion process. Not actually the dry digestion process, because in this sprinkling of water is also being given so that microorganisms can work properly or carry out the decomposition process in a proper manner. The organic waste stream is digested in batch mode, that is, once it is fed, it is left unattended into a simple garage like digester, as we can look from the diagram, having an airtight door. The material does not require to be transported once the door is closed. The term dry digestion, it is mostly taken or mislead that this process is to be played in the absence of water. The bacteria requires a wet environment involved in the anaerobic process, as I have already told, and are only active in the liquid phase of the substrate. So water should be added for proper functioning of the digester from time to time. Now the uses of anaerobic digesters. Anaerobic digesters, they are or their end products can be used for various purposes. For example, agriculture. In agriculture, animals and crop waste are typically used as a feedstock for anaerobic digesters, and energy produced can be used in the homes. Next is industrial. Organic waste from the food processing industries can be used as feedstock for an anaerobic digester, as it has 15 times the methane production potential than the dairy cattle manure does. During the process of food waste, substrate may also be combined with manure to improve the methane generation in the process. Wastewater treatment plant. It helps to digest sewage sludge in anaerobic digesters and eliminate pathogens in the wastewater. Some municipalities have even begun to divert waste from landfills to the wastewater treatment plants. This relieves waste burden placed on the local landfills and allows for the energy production. Anaerobic digesters make several contributions to the climate change mitigation. Digesters collect biogas or landfill gas that would have been emitted anyway because of the nature of organic waste management at the facility where the digester is in operation. Another benefit of anaerobic digester is the displacement of fossil fuel based energy that occurs when biogas is used to produce heat or electricity. Now I am taking UASB reactor, that is upflow anaerobic sludge blanket reactor. Upflow anaerobic sludge blanket reactor, it is the technology which was developed by Lettinga and his co-worker in the late 1970s. Originally, the reactor was designed to treat industrial wastewater, but later its applications was extended to treatment of sewage. Nowadays, the use of UASB reactor is extensively for the treatment of several types of wastewater, forming a part of high rate anaerobic technology. Now, in this slide, the outlay of UASB reactor has been shown. The UASB scheme basically consisted of influent tank, a parallelistic pump, cylindrical UASB reactor, gas liquid solid separator, which is also known as GLS, effluent outlet, gas outlet, and a collection system. In this, the functioning of UASB reactor has been explained by showing in the diagram. A UASB reactor is basically a tank that has a sludge bed in which organic fraction of the wastewater is degraded, and as a consequence of this digestion, biogas is produced. The success of UASB reactor lies in the development of dense sludge bed at the bottom of the reactor, which is formed due to aggregation of suspended solids and bacterial population into flocks and granules. The influent is pumped from the bottom of the reactor by parallelistic pump. The wastewater comes in contact with the microbial sludge bed and gets treated, and the treated water is collected from the top. At the upper part of the reactor, a GLS separator is kept above the sludge bed, due to which a blanket zone so formed where some particles of biomass are suspended. This zone acts as a separation zone between the water flowing up and the suspended biomass. Now in this slide, factor affecting the performance of UASB reactors has been explained. First, we will take organic loading rate. Organic loading rate is an important parameter that considerably affects the performance of UASB reactor. Higher values of OLR can cause reduction in COD removal efficiencies in wastewater treatment system, and higher loading rate could cause unrecoverable acidification, suppressions of the methanogenic activity due to serious imbalance between methanogens and the acidogens, as well as inhibition of methanogens by volatile fatty acid production. Next is nutrients. The growth of anaerobic microorganisms depends on the availability of essential nutrients like nitrogen, phosphorus present in the wastewater. Deficiency of these nutrients could negatively affect their growth of microbes and also efficiency of anaerobic degradation. Next factor is hydraulic retention time. The hydraulic retention time is defined as the average time that wastewater spent inside the reactor. HRT is affected by both the flow rate and composition of wastewater entering the UASB reactor. High HRT increase the contact time of the wastewater with the sludge and hence improve the effluent quality and biogas production rate. Therefore, a suitable HRT is very important for proper wastewater treatment in a UASB reactor for better treatment efficiency as well as quality and quantity of biogas concentration. Next factor is volatile fatty acids, which are important intermediate products in the formation of methane. The right concentration determines the efficiency of substrate removal from the reactor. Sudden variation in HRT and OLR could cause the accumulation of VFA and stressful conditions in the reactor during the breakdown of complex organic matter. Therefore, VFAs should be monitored and parameters adjusted in order to avoid their accumulation in UASB reactor to prevent the inhibition of methanogenic organisms, thus reducing biogas production. Now next, another important factor is operational temperature. In anaerobic digestion process, operational temperature greatly affect the biodegradation and biogas production rate of any anaerobic reactor. The temperature at which anaerobic reactors could operate include cryophylic, that will range from 0 to 25 degrees Celsius, mesophilic, the temperature will range between 25 to 40 degrees Celsius, and thermophilic conditions. Here the temperature will range between 40 to 60 degrees Celsius. Another factor, that is operational pH. It is also a very important parameter for anaerobic digester, as microbes are highly pH dependent and require suitable conditions of pH to grow optimally. pH far below or higher than the required range could cause an accumulation of acetate, thereby inhibiting the methanogens and leading to conversion of COD to volatile acids instead of methane production. Therefore, mostly anaerobic digestion reactors have been operated at pH between 6.5 to 7.5. The standard operating method to keep the pH in this range has been found to be the addition of lime and bicarbonate salts, or by reusing treated effluent in the reactor. Now, advantages of UASB reactor. It is having high efficiency. Even at low temperature and high loading rates, it can work efficiently. Its simplicity, its construction and operation of the reactor is quite simple. It is also flexible. The technology is very flexible and can easily be applied on either a very large or a very small scale. Low space requirement is, as the height of the reactor can be increased or decreased according to the requirements. Low energy consumptions, as heating of the effluent is not required to reach the working temperature and all plants operation can be done by gravity, the energy consumption of the reactor is almost negligible. Moreover, energy is produced during the process in the methane formation. It also produces sludge in low concentration. The production of sludge is very low as compared to other aerobic treatment processes, and it is due to slow growth of anaerobic bacteria. The sludge produced is stabilized for final disposal with good dewatering characteristics. The left sludge may be stored for longer period of time even without a notable reduction of activity, and can be used as an inoculums for startup of other reactors. A low nutrients and chemical requirement is there. Generally, sufficient and stable pH can be attained without addition of any of the chemical in case of sewage. Macronutrients, that is nitrogen, phosphorus and micronutrients are also available in sewage, and at other side toxic compounds are also absent. With the advantages, there are also some disadvantages of this UASB process. For example, low pathogen and nutrient removal is there. Only partial removed except for helminth eggs, which are effectively captured in the sludge bed, are removed. Nutrient removal is not complete, and that's why a post treatment is always required in this process. This process takes long startup due to the low growth rate of methanogenic organisms. The startup takes longer time as compared to aerobic process when no good inoculum is available. Possible bad odors. Hydrogen sulfide is produced during the anaerobic process, especially when there are high concentration of sulfates in the effluent. A proper handling of biogas is required to avoid this bad smell. There is always necessity of post treatment in this process. Post treatment of the anaerobic effluent is generally required to reach the discharge standards for organic matters, nutrients and pathogens. So, these are some of the disadvantages of UASB reactor. Now, to conclude the lecture, anaerobic digester, they are very efficient and economical point of view, they are very good, but if they are used in a proper, or if the energy so being produced is used in a very proper manner. They are very cost effective also, anaerobic digester, because they require very small land area for construction. In the maintenance also, there is very little maintenance is required in the reactor, plus it is very easy or very smooth in operation. On the other hand, the organic waste or organic load is efficiently removed during the process, and the energy produced in the form of biogas, which is a very important part of these reactors, if it is used effectively and efficiently, can produce fuel to be used for various purposes. Therefore, these kind of digesters can be used for small commodities where the population load somehow increases during the tourist season or during the rainy season. Above all, the treated water which is being generated from the treatment process, it can be used for various purposes. For example, we can use in our flushing toilets where maximum of our water gets wasted. Other uses can also be there, like we can use it in our car washing. The water can be used in horticulture. The water, it can also be used for gardening purpose, etc. So overall, if we see, it can reduce, or it will definitely reduce the load of our natural resource, that is our water, if it is being effectively and efficiently used.